Hello everyone, this is Shuai from Nanyang Technological University in Singapore. I'm very glad that I can present our paper, which is about robotic micromanipulation of biological cells with friction force-based rotation control in IROS 2020. This work was done by me and my supervisor, Professor Wei Tech Ang. There are multiple important biological applications where manipulating a single cell is a fundamental and crucial process. For example, the intracytoplasmic sperm injection, the embryo biopsy, and others. They all request that the egg or the oocyte should be immobilized first and then rotated to the designed orientation. As the data shown in the chart on the right-hand side, the total number of the associated clinical applications involving cell manipulation is keeping increasing over the last decade. Traditionally, in clinics, cell manipulation can only be performed by well-trained and skilled embryologists. However, manual operation may be lack of accuracy, efficiency, and reproducibility, especially for the rapidly growing demand for the associated techniques and applications. Therefore, some advanced manipulating methods are imminently called for. The technology and research about robotic micromanipulation have been widely investigated for several decades. Compared to the manual operation, the robotic micromanipulation is superior for some cases with better efficiency and operation precision. Generally, robotic micromanipulation can be divided into two catalogs, which are field-driven micromanipulation and the ways realized by the micromanipulator with different forms of the end effector. Some popular methods for the field-driven micromanipulation include optical tweezer, acoustic wave, and the electric field. The field-driven micromanipulation method usually have higher resolution for the control forces with the contact-free operation form. However, some unwanted effects have to be faced with for the field-driven methods. For example, the manipulating forces output by the optical tweezer or the acoustic wave are relatively small. On the other hand, the source field may raise some unknown or side effects for the biological cells. Like the electrical field, has been proved to be detrimental to the development of the biological cell. Besides, these methods are still relatively primitive and only capable of dealing with the 2D manipulation problem. Conventional cell micromanipulation methods for applications in the life science are commonly realized with the mechanical micromanipulator and the proper end effector. Robotic micromanipulation may provide an insight by automating these traditional manipulation methods. Common mechanical micromanipulator only has 3 degrees of freedom. Hence, operating the microobject greatly relies on the specific design of the end effectors. For example, the MEMS microgripper and the capillary micropipette. They can provide larger output force to hold or grasp microobjects. Actually, micromanipulation based on the capillary micropipette is currently the most widely used and accepted method, aided with some specifically designed devices or several micropipettes with different dimensions and forms in cooperation with each other. Biological cells can be manipulated in many scenarios. Compared to the MEMS microgripper, the micropipette is less costly and can be freely replaced in avoidance of cross-infections in clinical practice. Therefore, automating the micropipette-based cell manipulation is promising to bridge the gap between the technology and the clinical applications. This is also the reason why we chose it to design our robotic micromanipulation method. Several methods with capillary micropipette and the micromanipulator have been explored to rotate the biological cell. For example, for an oocyte fixed by a holding micropipette, a beveled micropipette can rotate it through the poking force. However, the weak control of the vacuum pressure may cause the oocyte to escape or hard to be rotated. In addition, the sharp tip end of the injection micropipette still has high risk to cause traumatic damage to the oocyte. Friction can also be directly utilized as a driving force for cell rotation. Aided with the assisted device shown here, the cell rotation can be realized as designed. Nevertheless, it may cause some occlusion during the autoplane rotation, which may affect the precision of the visual servo control. At the same time, the compression along the Z direction cannot be tuned or controlled. Compared to the existing method with micropipette, our method has advantages that it doesn't need to precisely control the vacuum pressure and only needs the standard clinical setup without any additional devices. The non-invasive manner will neither cause any physical trauma to the cell nor any occlusion for the vision algorithm. Furthermore, we quantitatively analyze the effect of the compression for the cell rotation, which helps the system to rotate the cell more stably. Our cell rotation algorithm is proposed and validated based on the standard clinical setup, which is built on an inverted optical microscope. Since the egg, oocyte, or the embryos at the early developmental stage share similar morphology for the mammalian animals, here we use a model of the mouse oocyte for illustration. Polar body is a critical feature of the oocyte through its development process. Its polar position to the cytoplasm indicates the orientation of the oocyte. For a random oocyte, its rotation is commonly realized through two steps, in-plane rotation and out-of-plane rotation. The rotation axis of the in-plane rotation is perpendicular to the image plane, while it is parallel to the image plane during the out-of-plane rotation. To be specific, out-of-plane rotation is to bring the polar body close to the focal plane, while the in-plane rotation is to precisely adjust the oocyte to the target orientation. Based on this, we propose our micromanipulation method for in-plane and out-of-plane rotation respectively. The cell rotation is generated by the friction force. For the in-plane rotation, the injection and holding micropipette and the cell should be brought to the focal plane simultaneously first. Then the cell is trapped between the micropipettes. With the injection micropipette fixed, the cell will start to rotate with the moving holding micropipette. For the out-of-plane rotation, initially, the micropipette and the cell are also on the same focal plane. Then the holding micropipette will be lifted a certain height and then moves along the y-axis toward the injection micropipette until it presses on the oocyte. This ensures the firm contact between the oocyte and the substrate. However, to avoid causing any occlusion to the intracellular structure in the image plane, the height that the holding micropipette can be lifted is calculated and limited. After this, the substrate will start to move accordingly. The out-of-plane rotation of the cell is generated. Usually, the biological cell is not a perfect sphere. If the applied compression is too small, the micropipette may detach from the oocyte during the process of rotation. Hence, we tend to apply relatively larger compression on the cell with its micropipette. However, we find out that for both the in-plane and out-of-plane rotation, the compression has the maximum value in order to generate a valid rotation. To analyze the effective value of the compression, we assume that the oocyte is an elastic sphere and the micropipette are solid bodies. The force models are built as they are shown here. Epsilon here represents the compression applied on the oocyte. The subscription IHS means the corresponding compression for the oocyte in contact with the injection or holding micropipette or the substrate. Since the field of view on the microscope can only reflect the 2D information, the compression can only be precisely controlled along the y-axis of the system. However, with the force models, all the compression can be derived as the total compression along the y-axis is determined. Here, the total y-direction compression is controlled by adjusting the distance between the two micropipettes. The rotation analysis is then visualized in these two graphs, where the vertical axis represents the angular acceleration of the oocyte when it starts to rotate. The horizontal axis is the compression y. The shaded area here is the region for the effective rotation, where it means that the oocyte can be rotated with the movement of the micropipette or the substrate accordingly. We also notice that the value of the angular acceleration here is very large, which indicates that the acceleration process is instantly. We also notice that the value of the angular acceleration here is very large, which indicates that the acceleration process is instantaneous. Once rotation is generated, the oocyte will catch the speed of the moving micropipette or the substrate immediately. Out of this effective region, the oocyte cannot be rotated. Therefore, the optimal compression y can be some value in the middle of this effective region, which guarantees the rotation and leaves some margin for the oocyte who are not perfect spheres. Also, rotation analysis reveals that how much compression is good to generate rotation. The precision of the micromanipulation also requires that the orientation of the micropipettes should be calibrated. The micropipette is a capillary glass tube which has a bend part in the end of the tube. The micropipette is inserted to a pipette holder and the holder is attached to the mechanical micromanipulator. The calibration procedure is to adjust the orientation of the micropipettes until their bend part are parallel to the platform and also parallel to the x-axis in the image plane. The inclined angle to the axis can be directly read from the images, while the inclined angle that the micropipette is held is not able to be obtained just with the images. However, taking the micropipette shown here as an example, if we can first roughly adjust it to make it point upwards and locate in the fourth quadrant, then we rotate it with a certain angle clockwise, then the inclined angle can be known with the geometric relationship as it presented here. The cell orientation recognition is implemented by the split and merge method. The region of the outside is firstly localized by a template tracking method. Then the image patch will be split into many uniform superpixels. The superpixels are then merged into larger clusters through the spectral clustering method. These large clusters will be further merged based on their grayscale similarities until the contour of the intracellular structure appears. With this control, we can obtain the relative position of the polar body to the cytoplasm. Here we define the concave region as the region left after we extract all the points inside the intracellular structure from its convex hull. Then the main position of the centroids of the concave region will be treated as the point of the polar body. With this, the orientation of the cell can be determined. This example shown here is the one that the polar body is not on the focal plane and will not be detected by the proposed algorithm. Here we set a threshold for the area of the concave region. If the total area of the concave region is larger than this threshold, it indicates that the polar body of the cell is detected. Otherwise, the out-of-plane rotation will continue until the polar body is detected. Here is the control structure for the whole system. The half of the above is to align the micropipette, while the bottom part is to rotate the cell on the visual servo system. Here is a demo for the cell rotation with our strategy on a mouse outside. First, it is the out-of-plane rotation, which aims to bring the polar body to the focal plane. Then, the outside will be oriented to the design position with the in-plane rotation. To test our cell rotation strategy, we did an experiment with 50 mouse outsides. 10 of the outsides were used for checking the mechanical properties like the membrane's elastic modulus of the outside. We took 2 image captures for each outside and manually labeled the position of the polar body and also calculated the orientation of the outside. With total 100 outside images, the recognition algorithm successfully showed the correct orientation 96 times. Then we took 20 outsides for the in-plane rotation test. For each outside, we first manually rotated it under the microscope until its polar body was clear in the image plane and at the 9 o'clock position. After that, the outside was randomly rotated with the in-plane rotation. Finally, the system was asked to perform the in-plane rotation to rotate the embryo back to the 9 o'clock position. For each outside, we tested the in-plane rotation 5 times. With total 100 trials, the system achieved a success rate of 95% with a precision of 1 degree. As for the out-of-plane rotation, we took the last 20 outsides. Similarly, for each outside, the system tried the out-of-plane rotation to bring the polar body of the outside to the focal plane and repeated 5 times. The overall success rate reached 94%. Generally speaking, the experimental results showed that our cell rotation strategy and the control system is competitive for the task of micromanipulation of cell rotation control. In conclusion, we propose a micropipette-based robotic micromanipulation strategy for cell rotation control, which can be further utilized in some biological applications like embryo biopsy. Our method achieved a relatively high success rate in the experiments, both for the cell rotation control and the orientation recognition part. Besides, this work also establishes a deterministic model to manipulate the cell with clinical micropipettes under friction force-based rotation control. The following work will focus on the part that how to improve this micromanipulation method and how to make it more robust and efficient on the conditions with some uncertain factors. Thank you for watching.